Now jumping directly to the topic of challenging membrane proteins and the targeting towards the functional lead generation as well as characterization. We know many of the membrane proteins are hot targets against which the biotherapeutic teams are developing antibodies, whether these are antagonistic or agonistic ones. The key players are molecules like GPCRs, SLCs, ion channels, different kind of claudin isoforms, as well as CD3 complex. And the topic of this discussion today would be primarily on the CD3 complex, which is an immuno-oncology target. So this protein complex is very bulky. It has TCR in between, as well as on the flanking side are the CD3 heterodimer molecules. And intracellularly, there are zeta chains, which are for the signal transduction. It is a prime target for development of monospecific, bispecific, and multispecific antibodies, primarily for the activation of the T cells in immuno-oncology space. Traditionally, the heterodimers around the entire complex have been made available because the complex has been too challenging to be produced intact. And these two heterodimers are epsilon and delta, epsilon and gamma, and they need to be produced in such a way that there is no homodimer formation happening, precise subunit combination is happening, as well as stability and consistency of the produced product, and primarily used for the characterization of lead candidates. At Cactus, we provide these products, for example, not only from human, but cyno, mice, and different toxic species, and we carefully validate the activity data around these using the traditional OKT anti-CD3 antibody. And also, this is a slide showcasing how carefully we try to maintain the quality as well as consistency of the products that we make available. So CD3 heterodimers, as I mentioned, traditionally has been used and in use currently as well, but they pose a challenge. The problem is that when you use these CD3 heterodimers in a reductionist approach, you basically generate lead candidates against the immunodominant epitopes. And some of these epitopes are actually not exposed in the entire complex. And this leads to a problem that some of the leads that are generated early on in the downstream characterization moments, they fall out. They are not the leads anymore because some of the experimental results are not translating in the in vivo experiments subsequently. And also the antibodies generated normally against these hidden epitopes, they demonstrate good binding in vitro assays, but poor binding in the subsequent assays. Also, the antibodies are more likely to be not agonistic, but more likely they are antagonistic in nature. And the prime reason is that the TCR molecule is not present.